Okay, so hello. Now I want you to focus on this very nice animation. As it happens for most things in science and engineering, actually the inspiration, right, comes from nature. And this is no exception for artificial molecular machines. We also have biological molecular machines in our bodies, and this is actually how they look like. And all of them have something in common, right? So the nice thing about these biological molecular machines is that they work much like a train or something like a car. So they walk on these tracks, and they're able to transform chemical energy into mechanical work. And they do this by using a very nice molecule known as ATP via a process known as ATP hydrolysis. And even if you don't know much about biological motor proteins, what you could see through this animation is that they come in different shapes and different sizes, and they also have different functions. Also, motor proteins walk on oriented tracks inside the cell. Each of these different types of motor proteins have actually different types of tracks. And if you believe me for 1 second, and you believe that this is a very detailed description of a cell, just with a nucleus and a membrane, I want you to know what kind of tracks motor proteins use. So we have 2 different types. We would have actin, which is at the periphery of the cell, and then we have microtubules, which I also have here. They look like basically an hollow tube. And each of these 2 different cytoskeletal filaments have also different types of motor proteins. We would have myosins for the actin, and kinesins and dyneins for the microtubules. And all these are just names for you, right? But if you have seen the animation before, you would like to know how we actually study and see things in the lab. It's funny because we cannot see things like we see in an animation. For us, it much looks like an agway full of dots that walk on these on these tracks, and this is how we are able to study things. This is something happening at the nanometer scale, so we really need a microscope to see them. But in the context of artificial molecular machines, what we're interested about is really like mimicking functions. So to mimic function, you also have to know what kind of functions you're interested in. And for example, I've talked about myosin, I talked about kinesin, and of course, if you're a structural biologist, you know how the structure works, but if you're a general audience, you don't. So what I want you to know is that there are some elements that are really interesting to mimic. So for example, here I point to you a core element responsible for ATP hydrolysis, and also a relay element that is able to communicate between the core and the mechanical element. And this is actually how it works. So here I have a motor head of a kinesin, which is where the ATP hydrolysis magic happens. And what now my colleagues are going to show is, or answer the question, can we engineer these motor proteins ourselves?